All right, next is going to be a recap of this whole process because there's a lot of parts, okay? So here we have DNA that has been opened down the hydrogen bonds by helicase, which is the bright yellow wedge. The same thing would be happening on the opposite side as well. These black circles are the single-stranded binding proteins that are holding everything in place so this whole process can work. We are growing this. We are growing the strand, the replication process in this direction, but we would also be growing it in this direction at the other replication fork. Okay. If this is the 3' end of this nucleotide here, this would be the 5' end of this one. So obviously 5' and 3' would be at the other ends of this molecule like that. Now, DNA polymerase III is going to be replicating this, but remember, primase, which is not labeled here, which is unfortunate, primase would have to come in and place a little RNA bit here, because this is like empty space, and that's the only enzyme that can add a nucleotide to kind of empty space right there. So we're going to add that nucleotide. Its 5' end is this way. We know we can't add to that, but we can add to this, and we're going to grow this right on up into this ever-opening replication fork. So there's our primer. We're going to add nucleotides to that, and continually adding to the 3' end and go on up there. However, on this side, if we put primer here, look what's going to happen. Our 3' end is going to be going this way, and that's fine. I suppose we could grow, assuming there's DNA going this way, which there would be, we could grow it this way. But what about this region? I can't add to the 5' here. So if we're going to add DNA here, then I've got to put a primer in perhaps here and then grow out that way by adding on to this exposed 3' going this way. And I'll put a primer in here, and I'll put a primer in up here. And every time this opens up a little further, I'm going to put a primer in and grow this way, put a primer in and grow this way. But I have to wait for this to open. Because I have to wait for that to open, I end up with these little pieces. And then once I have those pieces, which are called Okazaki fragments after the guy that discovered this, they get knitted together. The way they get knitted together is an enzyme named DNA polymerase I comes in, takes out the RNA primers, puts in DNA, and then this enzyme, ligase, is going to make that final stitch to knit the fragments together. This is the leading strand, this is the lagging strand. Okay? Here's another picture. All right, we are having an origin of replication right here. This is a replication fork, this is a fork, and they are growing the strand this way and that way. Okay, so there's your lagging strand and leading strand. Of course, those would be opposite, going down this way, but there we are. You can see kind of how this is going to be working. Okay, on this side, the lagging strand's coming this way, and then leading going into this fork, and so on. All right, let's see if we... Oh, this is showing you actually primase coming in and putting down a primer, and then DNA polymerase going off of that. There's your primers and your DNA polymerase III building off of those primers. Okay. DNA polymerase I is being shown in this image, taking out the primer and replacing it with DNA. And then ligase is what comes in and makes the final little stitch so that they are all connected. And please note here, when we remove that primer, we can't add in any DNA there because there's nothing to add to. So this is going to shorten this chromosome a little bit, if this is at the actual end of the chromosome. And that can actually cause a little bit of a problem, genetically speaking, at some point for some eukaryotes, and it's called chromosome erosion. Okay? Basically what's happening is if you copy a cell over and over and over and over again, at the very ends of the DNA, there's going to be this permanently shortened piece. And if we do it again, it's going to get a little shorter and a little shorter. Now, DNA has some tricks to deal with that. First of all, there are little extensions of DNA on the very ends of your chromosomes that you need to think of, like you think of the little ends, protective ends on your shoelaces that keep them from fraying. These ends are called telomeres. And those telomeres are just kind of unnecessary nucleotides, but that are necessary because if they get chopped off, as in this process, we aren't going to miss them. They're not coding for anything. But once you've copied your DNA about 50 times, you eat through the telomere, and eventually you start getting into the good genes that you don't want to erode. And it's thought that may cause some of the genetic problems we see that we associate with age. Chromosomes do get shorter with every replication. So the telomeres are there to prevent that from happening, or not prevent that from happening, but prevent that from having a bad effect on us, at least for a number of years, a number of cell divisions. Okay? This is the last summary section. This is just a nice diagram showing you helicase working here to open up the zipper. Primase is working here to lay down RNA primers. Primase would have maybe been here before also and gotten this strand started. DNA polymerase 3 acts to actually elongate the strands on both sides, but it does so in fragments on this side, these Okazaki fragments. And ligase and DNA polymerase 1, these two enzymes have to work to knit those fragments together. This side that doesn't have that fragment problem can just keep continuously adding to this 3' end, but the other side has to do it in fragments, so one is the leading and the fragmented strand is the lagging strand. All right, that's your summary. Thanks for watching, and I will see you in the next podcast.